In this video, what we're going to be talking about is how to take a human cell count in a biochemistry lab. So before we get going, I just want to go over the purpose and the safety and sanitation measures. So the purpose is to show the steps involved in how to take a human cell count in a biochemistry lab, and then also show high school students the technology and chemistry and how they're interrelated, and what type of work is being done every day in a biochemistry lab. So throughout this whole process, lab safety measures and sanitation is taken into effect. So the first step in this whole process is removing the media by aspiration. This step occurs after the test tube containing old media and cells has been centrifuged, and it is important to remember that the tip of the aspiration tube should just be at the top of the liquid, and as the liquid is being sucked out, the tip continues to move down the test tube. Now, at the end of this process, the only thing that should remain is those human cells at the bottom of the test tube. Immediately after this step is done, the cells need to be resuspended in 1 milliliter of fresh media. After these cells are resuspended in fresh media, the fresh media and cells need to be mixed together to prevent those cells from drying out. Once the new media and the cells have been mixed together, 10 microliters of that cell suspension is going to be added to 10 microliters of trypan blue solution. Now, it's important to already have the trypan blue solution measured out and placed into its own vial. It's also important to think about what is happening here with this trypan blue solution. So the trypan blue solution is a chemical, and that chemical can actually be absorbed by live cells. And we'll get to that here in a minute. But after you've mixed together the trypan blue and the cell suspension, you're going to take that mixture that's just been produced and you're going to place it onto a slide plate. That slide is then going to be placed into this automated cell counter. So those live cells, and just the live cells, have absorbed that chemical, trypan blue, and as that's being done, once it's placed, the slide is placed into this automated cell counter. Those live cells that have absorbed that trypan blue can be detected through this machine. So after the machine goes through its process, it's going to output this reading. A couple things on this reading. There's a total cell count, a live cell count, and then a live cell percentage. So these live cells are the ones in this particular device that have absorbed that trypan blue. And because of the way this is set up, this device can detect that trypan blue. Now, what it does to get this live cell percentage is it takes this live cell count and it divides it by the total count. At the end of this process, the slide needs to be disposed in that biohazard waste receptacle. Little check on learning here. I just want to know what stain was used to mix with the cell suspension for this process. Answer: trypan blue. So I hope you've enjoyed this video. Outlined here below are the 8 steps to take a human cell count in a biochemistry lab.